This is a success story of an insectary that was set up in a rural setting at Yagaum that is located 30 minutes outside of Madang Town in Papua New Guinea. Through many years of malaria research in PNG, the insectary that was set up by Derek Charlwood in 1981 became the centerpiece of carrying out outstanding malaria research for over 40 years in PNG and the global community. The research generated from the insectary involves testing transmission dynamics in known laboratory reared mosquitoes and insecticide-treated test that is rolled out as a nationwide control intervention. This documentary presents the success story of the Papua New Guinea Institute of Medical Research Insectary.
Wala Sakel, we've been working on the entomology lab.
Hi, I'm Lisa Reimer, and I joined the entomology unit in 2009 and spent 4 years working with the team.
My name is Michelle Katuseme. I'm a senior scientific officer with the entomology lab at the PNGIMR vector-borne diseases unit in Madang.
Nimblomi Hega Sakel. Mi start work on the PNGIMR in 2010. Mi work as a lab technician.
My name is Lincoln Timinao. I'm currently a PhD student studying at the James Cook University and also working as a senior scientific officer at the PNG Institute of Medical Research.
In a long period of time, me no talking Pidgin, so I'm sorry, I have to speak in English. It was around 40 years ago that we attempted in Madang to start a small colony of Anopheles ferroti from Bilbil village. It took a little while to get it going because, as with all these situations, the mosquitoes didn't really want to mate. So we introduced tutor species in the form of Culex quinquefasciatus males to get the mosquitoes, the male mosquitoes, swarming and eventually mating.
Along 1983, Patricia Graves, Tom Bucket, Raymond Paru, itisimi na line me. Emi make him name talk, you look him na you behind me. How am I lining me long walk him kai long all first insta, second insta, na third insta lawe, one time fourth insta. How kisimol pupa, na how lo puti moli go inside, lo net, na how lo breedy mol can, so next round by come up. Time me adult mosquito come up, and me mas kisim na Larim only stop angry for 3 days. All get a mama mosquito pulling blood now, me rouse him. Now me put him go inside long. Me carry net want him mama mosquito, he go put him little sugar pad. Now he stop for 3 days. After 3 days, mama mosquito by pining water lay him egg. So me by go kiss him little this, bowl this, na cut him all filter paper. Put him lo this, na put him water now. Put him go inside lo net. So that mama mosquito by go lay him egg inside lo this. Me browse him the filter paper, he got egg, put him inside. Na first install by he go and by come up. So me must give him kai long all. Blow all first insta, me must give him kai. Na second insta time, me go second insta, and now. Me by try me walk him narap la kai belong all third na fourth. After lo des la third fourth Kai go, kai na only come a pupa now. Only come a pupa now, and me by rousing pupa lo tube. Me rouse him all, na me by put him inside lo one black cup water. Na me put him go inside lo net, na inside lo net, by all emerge. Na only fly that wing. Now me by walk him sugar water, put him on top lo net. So all after long 3 days, all female mosquitoes want a male mosquito. All crossmate finish. Me rouse him sugar pad, na larium all female by ilakissim blood. Desla by go na enough by desla colony by go round all same tasol. While me walk him desla, he got narapla next load walk lo come. So me must keep up one time desla all get a step by step. Now me by round he come back in. Colony belong mosquito and me must keep him. For how much years me walk him desla ego na and me experience finish long desla.
When I joined the unit, the first thing that we wanted to do was to establish local colonies of Anopheles punctulatus and Anopheles ferrauti. And colony establishment can be really, really challenging for some species. So mosquitoes need wide open spaces and they need, natural cues from the environment in order to mate and seek out a host and blood feed and lay eggs. And so we've ran into quite a few challenges at each of those stages. So we had to build really large cages to give them space to swarm and mate. But then the cages were so large that we also needed really a critical mass of mosquitoes in those cages. So everybody got involved with collecting larvae, identifying mosquitoes and putting the right species into the cages. And oftentimes we would have to supplement with artificial mating. So individually, hand mating each female in order to get that critical mass of mosquitoes in the cage.
Why did we create a new backcross colony of Anopheles ferrauti? So in 2012, we realized that our Anopheles ferrauti colony mosquitoes were maintained for a really long time and could have probably lost the ability to successfully carry malaria infection, or they were refractory to malaria infection. So led by Dr. Lisa Rima, Hega Sakel, Walpuma, and myself did a series of experiments to create a new backcross colony of Anopheles ferrauti. So we did a couple of experiments to do this, to backcross the Anopheles ferrauti colony that we have here at the Yagaum with wild Anopheles ferrauti from Dogia and Maraga area in Madang, the south coast of Madang. So these experiments, we did a series. We first got wild larvae of Anopheles ferrauti along Dogia and Maraga, and we tried to breed them with colony larvae here in Yagaum in cages. However, we found that the breeding rate for them to naturally mate in the cages was really low, so it was unsuccessful. And this we determined by doing spermatica dissections under the microscope to see whether or not there was mosquito sperm present in the spermatica or not that indicated successful mating in a female mosquito. So we found out that it was unsuccessful to breed them directly in cages. So we decided to do another experiment where we collected wild larvae from the Maraga and Dogia area. Again, we brought them back, reared them until they were at the pupa stage, and then using the microscope, we separated the males from the female. So female pupa and male pupa was separated into different cups, and then we did the same for the our colony Anopheles ferrauti mosquitoes. So then we had two cages, where one cage had wild female pupa and colony male pupa, and vice versa. The other cage had colony females and wild males. So then we let them emerge, in the cage, and they became adults, and then waited to see if they successfully and naturally mated within the cage conditions. Initially, it was low mating, natural mating, so we supplemented the reproductive rate by doing artificial mating, where we got adult male and females from each respective cages and did force them to mate to increase our rate of the number of females that were mated. So this was done from January 2012 to March 2012. So by early March that year, we successfully had our first generation of eggs from these two cages. From that experiment, initially when we did the experiment, we were hesitant that it would not be successful. But once they quickly adapted to our insectary conditions and protocols, we had no doubt that we had successfully created the new Becker's Anopheles priority 1 mosquito colony here at PNGIMR.
Now that we've got all the adults in these huge cages, they need humans to feed on, and they only feed at night. And so we had to bring these cages home on the bus from Yagaum and blood feed them every single night, sticking 2 legs and one in each cage, sitting in the dark for hours in the evening, trying to get them to feed, really nurturing them through every single step of the way. Everybody in the team was really invested in these colonies, and eventually we were able to get them adapted to artificial feeders, and we were also able to set up the insectary so that we had reverse day-night cycles, so that we didn't have to work with them through the night, round the clock. The insectary was simple, but we were able to achieve appropriate containment and also a stable environment, stable climate for our experiments, using local materials and a bit of creativity. And I really felt like there wasn't a limit on the work that could be done in that insectary. And before too long, the team were conducting experimental infections with filarial worms and with malaria. And this is work that requires really strict protocol adherence and controlled experimental conditions. But some excellent work came out of the unit and continues to. And I think that personally, the infection work that I was involved in really has been the highlight of my own research career.
My PhD project was basically looking at the transmission of malaria parasites from humans to mosquitoes. And in order to do that, I needed a large number of Anopheles farauti mosquitoes. So in 2015, we improved the protocol, the rearing protocol, so that we could maximize the numbers, increase the number of mosquitoes that we produced, and also worked on trying to improve how the mosquitoes were being fed and the little conditions surrounding the feeding of these mosquitoes. And we have successfully improved our rearing capacity, and the largest number of pupa that we collected in a day was 9,000, which is a record for us. A really nice setup here where we can successfully infect our mosquitoes with Plasmodium vivax parasites, and that puts us in a very unique position to currently collaborate with other scientists around the world to test potential transmission blocking vaccines or antimalarials that can inhibit the growth of parasites within the mosquito.
And thinking back on my memories of the insectary, I'd say there's 3 things that stand out. One is the expertise of the technical staff, especially Wallaf, who knew exactly what the mosquitoes needed at any given time, and our collectors who knew where to find each species of mosquito, both larval stages and adults, and their skills with identification.
Namel time iga sampla kain or University Sumatin long overseas Yagaum also like go round only looking experiment long along colony finish na mi plabay go out miximoli go out lo how mi by go collecting all healthy larvae and mi play mi sa behind him road one cup na mi sa kisimol o igolo one site blow IMR ento isa go about by play go ties kisimol they come na by only make him experiment long only.
Second would be the enthusiasm of the early career researchers who joined the group. I remember some of them would come sneak into the lab late at night just to watch the mosquitoes, see what they're up to, and watch their behaviors. And I think that this enthusiasm, as well as some diligence to really optimize new protocols, helped move the expertise within the group forward.
Insectary one interesting building where mi amamas lo staff na lukauti mosquito na maintaining mosquito colony where interesting lo you lukauti mall challenges sa come up or lawisa die sampla lawisa die mi plaza sort lo mosquito because mi plan no got enough lawi lo growing mo lo collecting pupa na si mosquito that's all plus anyways lo how ba mi pla make him lawi go planting na kisim back mosquito la lookout team Anopheles one them mixed colony so mi pla blood feed lo mosquito direct feed lo mosquito lo cages lo sim beds egg beds na maintain him desplatupla colony inside the insectary.
As all of you probably know, insectaries provide a great deal of useful information because the insects in them are known and can be known, their genetics can be known, and their resistance status to insecticides can be known, so that they are, for example, useful for bioassays or testing new insecticides. They're also very useful for doing xenodiagnosis, providing information on mosquitoes that feed on people who are asymptomatic from malaria, say.
As part of my PhD, I was interested in understanding the contribution of asymptomatic infections to our mosquitoes, and hence I have brought our colony mosquitoes to the field where we allowed 30 of our mosquitoes which were prepared in a cup to directly feed on healthy individuals within a village, just to see if they would transmit malaria parasites through our mosquitoes. And so far we have established that asymptomatic transmission is ongoing in our villages, and more needs to be done to fully understand how much is being transmitted in order for us to really look towards eliminating malaria within our country.
It was really just a small enterprise, but as they say, from small acorns, large oak trees grow. And so I was very pleased to hear how successful the insectary at the PNGIMR has been over the last many years. So, very well done, and my congratulations. Many more years of work.
It's the excellent science that was produced collaboratively by the team. I think that, research that's conducted using local species has been transformative in informing, vector control policies in Papua New Guinea. Papua New Guinea mosquitoes have significant diversity, very different behaviors and susceptibility than other, global strains of mosquitoes. And, because that's different, any approach to malaria control, filariasis control, arbovirus control really requires research on those local mosquitoes, because the solution for PNG is not going to be the same elsewhere. And, I've definitely seen PNGIMR entomology unit really leading the way and producing really exciting research from the insectary. And with these new facilities coming, I just can't wait to see what comes next.
Despite the challenges posed by the ongoing effects of COVID-19, successful progress has been made on the Natnat project here in PNG. Natnat aims to build a framework in PNG for the rapid assessment and adoption of novel vector control tools in PNG. This insectary, also known as Belna Natnat Centre, is one of the first objectives of Natnat to strengthen laboratory, semi-field, field capacity to test new vector control tools in PNG. As you can see, right now, construction work is still ongoing. Thus, we are aiming to complete this project by the end of 2022. Nothing here can be done without high highly effective partnerships from our Product Development Partnership Team, the IVCC Indo-Pacific Initiative, led by Fred Yeomans, the Rotarians Against Malaria Team, led by Mr. Tim Freeman, the National Department of Health Team, led by Dr. Melinda Susaku, the National Malaria Control Program Team, led by Mr. Leo Makita, Belna Institute Team, led by Dr. Liam Robinson, James Cook University Team, led by Dr. Stefan Kal, Papua New Guinea Institute of Medical Research Team, led by Dr. Moses Laman, National Department of Works in Madang, and Ella Builders and Contractors in Madang. As you can see right now, construction work is ongoing. Thus, we are aiming to complete this project by the end of 2022.